And to show you more detail, here's another fluorescent nucleus stained. They use two different types of antibodies. One antibody stains heterochromatin. That's the DNA that's packaged, not used. The antibody used has a red fluorescent tag. The heterochromatin is always around the periphery. That's the DNA that's not packaged for that cell. The green fluorescently tagged DNA is the euchromatin. The DNA is kind of unwound and accessible for replication, transcription. And this kind of shows you how it's packaged. So here's the nuclear envelope. There's the laminin proteins. And the DNA is actually attached to the laminin proteins. Here's the heterochromatin all packaged up. Here's the more loosely dispersed euchromatin here. That can be accessible to like transcription enzymes, replication enzymes, things like that. So the architecture of the DNA in the nucleus is actually organized. And in different cells in a multicellular organism that use different genes, different parts of the DNA would be in this open region here, accessible. Right? And in different tissue, if those genes weren't necessarily be packaged up here. So each cell, each tissue uses certain genes, it organized the DNA in its own unique way. So it's pretty amazing. It's not all just jammed in there like string in a golf ball. It's highly organized. Okay, let's see to chapter 14. Faith, mother. I was going to say take somebody else, but I don't want to get in trouble. So let me kill this slide. Let's begin chapter 15. Let's see now how DNA is expressed in protein. Okay. This is a chapter on that's a description of the central dogma of life, which is summarized by this flow here, this phrase here with connected with arrows, that the information of biology always moves in this direction. Although we have seen it can go in the reverse direction, reverse transcription. Okay, so with Mendel's description of particulate inheritance, the 20th century journey to understand the chemistry of the inheritance, people began to also want to know, how does these molecules actually build traits? How does a phenotype result from particles of inheritance that you inherit? And the first clue was reported in 1908 by Dr. Archibald. What a name. I love that name. Anybody named Archibald anymore? If I ever have another kid, which won't happen, I get a new wife. That ain't gonna happen. I name him Archibald. Archibald Garrett. I love that name. Archibald. Hey Archie, you're bald. He was studying a disease called alkaptonuria. And alkaptonuria is an inherited condition, passed from parent to child. I think it's autosomal recessive. Okay? It's the symptoms are your cartilage turns black. So this is most of the white of the eye is I'm not cartilage, connective tissue. Here's white here. Here's the connective tissue in the ears. Kind of gets this black. It turns. Did I say white? It turns black. That's black, not white. And your urine turns black. Ooh. Okay? It's because of a defect in a certain enzyme. Okay? The black is the production of something called homogentisic acid. Okay? Which is a breakdown product of a couple of amino acids. I think it's tyrosine and phenylalanine. Yep. And there's another enzyme that then tends to metabolize homogentisic acid. But if the enzyme's mutated, it's not metabolized and it builds up. It's an enzyme called, I want to write it down, homogentisate oxidase. The gene for that, if it's mutated, means that this can't be converted and these symptoms result. And he figured correctly, he concluded that since it's inherited, this lack of enzymatic action, that genes must code for enzymes. That was his conclusion. One gene, one enzyme. And of course, enzymes are proteins. Does that make sense? I mean, he understood the biochemistry of this disease, that it was lack of a functioning enzyme. The fact that it's inherited means that the gene for this enzyme is not working. That's what's passed from parent to child. So a gene must code for an enzyme. Simple. Does that make sense? Okay. But that work wasn't widely recognized. But it's the first indication that of what genes produce. But the work that really caught people's eye was done in the 1940s by, I believe, two guys at Stanford, Beadle and Tatum. They were studying a fungus called Neurospora crassa. It's the orange bread mold. It's like a orangey growth on your bread. Okay? And they were doing classical genetics. Now, I mentioned this before, what classical genetics is. I'll mention it again. You want to study genetics in the classic way. What you first do is, one, generate some mutants. For Donald Trump, that means have kids. Right? For Beadle and Tatum, they would X-ray the fungus, produce mutant fungi. Okay? Then you would perform some sort of a screen to identify the mutants. In the fungus, what you do is you look at what they require to grow. What kind of nutrients do they require to grow? Like the wild type, the normal fungus, a normal healthy organism that's got no significant mutations is called the wild type. And your parents say, you're too wild. You say, that's good. That means you're normal. This is, for the fungus, this means the fungus could grow on minimal media. What they abbreviate the slide as MM. It means the media had like a sugar, some salts, maybe one vitamin. They could make all the rest synthetically. They had all the enzymes necessary to make all the other macromolecules they need. All the other enzymes and vitamins and nucleotides and all that stuff. But after producing mutants with X-rays, they produced some. The mutations would produce what are called auxotrophs. Does that bug anybody? The door open and close? I can close it. Doesn't bother me. Either that or spirits are walking in. Okay. You never know, right? These are auxotrophic mutants. That means they cannot grow on minimal media. They need a supplement. I think they screened thousands and found like 3 or 4 mutants. Okay? Then you try to understand the mutation, you do some detailed biochem. Now, what really brought this project to fruition was a postdoc and a grad student in the lab at Stanford, Zerb and Horowitz. They had 3 auxotrophic mutants of Neurospora. They were mutants for producing the amino acid arginine. They couldn't make arginine. Okay? Somehow in their X-rays, some gene involved in the synthesis of that enzyme was screwed up, and they couldn't survive unless you put them on an arginine diet. So here's the wild type. They grow on minimal media, and so they would grow. So if you have nothing in the media except the minimal media, the wild type grows, but none of the mutants. You see that? You can see the orange growth there. Okay? These are 3 auxotroph mutants. They have mutant 4, mutant 2, mutant 1. Okay? Now, if you add ornithine to the minimal media, now, okay, these are some intermediates in the synthetic pathway toward arginine. It's a multi-step pathway. Each pathway from one substance to the other requires an enzyme in a metabolic chain, okay, in the metabolic pathway. So you convert ornithine to citrulline, and then that's via an enzyme. Then citrulline is converted to arginine by another enzymatic reaction. Okay, the wild type can take the raw material, which I'm not sure, I don't remember what that is, and can use that. It can grow. It doesn't need any of these supplements here. It can make the arginine without any addition to minimal media, which is just sugar, salt, and like biotin. Now, mutant 4 on the minimal media couldn't grow, but if you had an ornithine, it could. And if you had a citrulline, it could grow as well. If you had an arginine, of course, it could grow. Okay. Mutant 2 couldn't grow in the minimal media, this auxotrope. Ornithine didn't help it either, but citrulline and arginine both allowed it to grow. So the mutation was a different step in the metabolic pathway. Right? And mutant 1, again, this auxotrope couldn't grow on minimal media, nor with ornithine added, nor with citrulline added, only if arginine were added. This mutation is happening later in the metabolic pathway. Okay, so these 3 different mutants, okay, we're talking about a chain reaction where we have one substance is converted to another, converted to another, converted to another, and in each reaction there's an enzyme. There's enzyme 1, there's enzyme 2, there's enzyme 3. And each of these auxotropes has a mutation at a different enzyme. Right? Okay? And their conclusion, you can read it on the slide from your textbook, each of the 3 arginine mutants showed a different pattern of growth in the supplemented minimal media. Sherman-Horowitz concluded that the biosynthesis of arginine occurs in a series of steps, with each step controlled by a gene that codes the enzyme for that step. This confirms the one gene, one enzyme hypothesis of Beadle and Tatum. Okay? And so you can see that the ARG4 auxotrope, the mutation was in that first enzyme. Because if we added ornithine, it could grow. Okay? The ARG2 mutant, the mutation was here. You had ornithine, it didn't help. Because the mutation was here. You had to add citrulline and arginine, then it could grow. And the